Hello everyone, my name is Sanam Soifar. Now today I'm going to show you how we can make a spheroid based on liquid overlay method with human mesenchymal cells. I have cultured the cells 3 days before in the 75T flask. Now I'm going to trypsinize them, make them single cells. After counting them, I will divide them between the wells which are covered before with the agarose so that they will have a U shape that will improve the cells to aggregate and collect in the center. Afterward, we will centrifuge the cells, and that will also be a positive thing to help the cells to come closer to each other and improve the aggregation process. After centrifugation, the cells will be incubated in the incubator under the standard condition for around 3 to 5 days. And afterward, we will see the round circular spheroid which we can stain them or do further experiment with them. Here is a normal lab which we use all the time for processing this kind of experiment. We need one bench with the normal pipettes, tips and suctions and stuff which you see I have for her almost prepared for doing the experiment. Then the second thing that we use is a normal centrifuge. Here you can see we will put it in a room temperature, that means 25 C, and that will be centrifuged at 250 RPM for 5 minutes, as I have set it here. And the last thing that we are going to use is the incubator. That is an incubator which we culture our cells in the lab. That is in 37 temperature and 5% CO2, which is a normal culturing cell condition for it is needed for this experiment. So let's start the experiment. Here, that is the mesenchymal cells, which I also prepared for you. They are one week in the culture, and now they are more than 70% confluent, so that we are ready to trypsinize them and go further. Here is another microscope which gives us chance to look at it with the camera. I will show you further here how do the cells look like. They are totally confluent. Here, as you can see, they are our mesenchymal cells. They are almost 100% confluent. That means they are ready to be used for our experiments. So from the beginning, we should do the process of trypsinization. That means I will collect this medium, that is the old medium which they were cultured in it. We will get rid of that. We will wash our cells once with PBS. Then we will treat them with trypsin. That will be kept in the incubator for 2 to 5 minutes. That depends on the cell types. After that, after checking under the microscope, the cells will be singles and roundish and swimming in the PBS, in the trypsin. Then we will collect them and count the cells, which is one of the important steps of preparing a spheroid. Here it is AAA, that is another type of that is used for trypsinization. There are different kind of elements, acotase, trypsin, AAA, different components which can be used for the same seal. Be careful that I have cleaned everything which is under the bench with the ethanol 70%. So before starting, be sure that they are clean. As you see, I collected once like this. Then we need to to wash it once with PBS because our medium contains FPS, and FPS will interrupt the AAA function. So we wash it once, just 30 seconds, and then go further. Here is PBS, which I use for washing my cells. We will use the normal 1000 milliliter pipette. That is 2 milliliter is enough for this issue. You see, I will throw it so slowly inside, like this. Then you will try to move it all around and cover it, the complete surface of your flask, to be sure that it is washed properly. And we get rid of it. Now, for the big flask, I will use 3 milliliter AAA, and that will be incubated for 2 to 3 minutes in the incubator. So again, move it and be sure that it covered almost, look, that is covering the whole surface. That means it is enough. So now we will put this in the incubator and wait for 1 minute. In between, I would like to show you the plate which I have prepared with agarose yesterday. That is a normal 96 well plate which I covered yesterday with the 1% agarose, low melting agarose. If you have a chance to see it, the wells, so you can see that the wells are having a U shape, so that help the cells to aggregate better. You should prepare it at least 6 hours before starting your experiment. Put it in the fridge so that the agarose will solidify, and after that you can use it for your experiment. 1 minute is finished. Now I'm going to check this, to see if the cells are singles or not. You will take it out. Be careful that the solution shouldn't come to top of it and shouldn't touch this head of it. You can see. You will hold the plate like this and tap it to the side. And here, see, these what are swimming in the medium, in the trypsin, is the cells who are detached. But we can also check under the microscope to be sure it is enough or not. As you can see, the cells are single, around, and swimming in the AAA. That means they are totally ready to be used for the next step of our experiment. So, for this one, first of all, we need to deactivate our AAA because that is for the cells dangerous to be treated for a long time. So, for this purpose, if you can remember, I add 3 milliliter AAA. Now I'm going to add 3 milliliter PBS to this to inactivate it. If you decrease the concentration of AAA, that will be deactivated anyway. So, don't forget to clean your hands every step to avoid any contamination. So, here is our PBS. I will add 3 milliliter. mix it a little. So now I need to centrifuge it to get rid of AAA and have a pellet of the cells as a pellet at the end of our falcon. The first thing is that you will write what it is. Then you are going to collect it and centrifuge it. Can pull it to the side, actually somehow like washing the pellet to be sure that you don't miss any single cells. So now I'm going to centrifuge this cell suspension to have a pellet of cells. For this one, we will use our centrifuge. The first thing that we need is the tara, which is the equal amount of water we have prepared here. In order to be able to do a centrifuge, and here we put it here. For this issue, we don't need 5 minutes. It is so much. We can do it with the 3. The rest stay as it was, and then we will press start and we'll wait for 3 minutes to be sure that the cells go down and we can divide it from each other. So now I'm ready. Press it. We'll open it. So here, as you can see, that is a pellet of cells. That is a huge pellet. So that means that we have enough amount of cells that we are ready to work with it. So come back under the bench. First, we are going to discard the supernatant. We will discard the supernatant, then I will add 2 to 3 milliliter PBS and solve the pellet in it, and then become ready to count my cells. For counting, we need 1, 2. This step is so critical because if you didn't pay attention, that can happen that you will get rid of your pellet as well. You will hold it like this, and you will approach it step by step close to your pellet, but don't approach so close. It will be okay if you let a little of your supernatant here instead of lose your pellets. So we will go in and start to get. So from here you should decide how much you can approach it. I keep it like this. It is enough, and we have the pellets. So now I will add of PBS and try to solve my pellet in it. As you can see, it is quite so much. So now I will get 100 microliter of my cell suspension and put it here as a sample, which I can work with it further for counting the cells. The that is a palette that we use for staining with trypan blue. Here is a trypan blue, 2% trypan blue. I will bring them down under the bench, but we should keep in mind that they are not sterile. But it's not so dangerous for ourselves. Before starting that, we need to prepare Neue Bauerkammer. That is what we use for counting our cells. I show you in the beginning. Now I'll show you how you should prepare it before starting anything. Here, that is a Neue Bauerkammer that contain of 2 parts. The first one is this, and if you can come closer, there are two lines on it here. You can see that it is more better shown in the PowerPoint that I prepared for you. Here is a place where we are going to count our cells. That is the most important part. We have 2 of them, one on top and one here. And the second part of it is this glass object. We'll do it all the time like this. Then you will put it on it and press it for some seconds, and then you see it is sticking on that. So it's now ready to use. It's hard nowadays with the mask to prepare it. So we have a specific dilution for trypan blue. In this case, that is 1:5. That means we will have 40 microliter trypan blue, which is mixed with 10 microliter of your cell suspension, and then we will load 10 microliter of that inside of it, and then we will go to count our cells. So, as I said for the beginning, here it is 40. We will have it in one well. I will make 2 to be sure if anything goes wrong. So then from our cell suspension, that 100 microliter that I have put here, we will take 10 here. Be sure that you have mixed it before gently and properly. Then you will go in, pipette it up and down, and then take 10 microliter of this cell suspension and go here in the wells and mix it gently and properly with our trypan blue. And then you will take again 10 microliter, and here you will hold it like this, support your pipette with this, with your other finger, and then try to load it slowly in the between your glass and your Neue Bauerkammer. After that, we will go to the other microscope and see how do they look like, and I see if there is any blue one as a dead cell that we can